Okay, shall we start? You are the earlier risers. Okay, thanks for getting up so early for the lecture. We're going to learn about memory stem cells today. Scientists have been interested in memory stem cells for over half a century. And at each time, there were bursts of discoveries following the emerging of certain technologies. And today, I will take you through the history of mammary gland research, to start at the very beginning when people started to become interested by memory stem cells. And I'm also going to touch on breast cancer stem cell theory. I think you have heard of before. Why mammary gland stem cells are interesting? They are interesting for many different reasons. First, mammary gland make milk, and improved milk production is an essential part of the economy. So it's of a great interest for agriculture industry. And we also know now that people think breast cancer might derive initially from stem cells or memory stem cells. So a lot of efforts are being put into the study of memory stem cells to identify the markers and to identify the mechanisms so that people want to target the so-called cancer stem cells. More importantly, memory gland in general is a very, very good model for biological studies. I'm sure you still remember these photos, right? In your physiology, do you remember? How did I describe these images? 4 seasons, right? We see start with spring, some flower buds appear, and summer with flower, and autumn lots of fruits, and winter is a bit bleak, but it's going to start all over again. So what I'm trying to say is that mammary glands undergo cyclic changes during the estrous cycle and during pregnancy, such that undergo growth, a rapid phase of memory growth during pregnancy, functional and structural differentiation during lactation, and then evolution that is associated with apoptosis, memory gland remodeling, and regeneration of the memory gland. So it's a very, very useful model for studying these cellular processes. Okay, so for a start, I want to just briefly revise the structure of the mammary gland, as you remember. So mammary gland A is made up of 2 components. No, sorry, it's wrong. Breast is made up of 2 components. The mammary gland, shown here, and the surrounding stromal tissues. The mammary gland consists of a system of branched mammary ductal tissue, and the end of it consists of the lobules, alveoli, that make up lobules, which then form lobes. Okay? And this whole structure is embedded in the memory fat pad. And there are 2 very important type of cells that are essential for the understanding of memory stem cells today. These are the memory epithelial cells, memory myoepithelial cells. And memory myoepithelial cells surrounds the memory ducts and memory alveoli. So it's at the basal side. You have gone through breast cancer, haven't you, right? Did you have breast cancer Have you taken cancer biology? Yes or no? You have. So you understand that basal breast cancer is more aggressive, right? So these myoepithelial cells are basal cells that surround the the basal side of the alveoli and the mammary duct. Whereas the luminal cells facing the lumen, that is either lumen of the alveoli or the lumen of the mammary duct. So it's these two types of epithelial cells that make up the mammary gland. Okay, so to recap, remember that mammary gland is derived from the ectodermal layer during earlier embryo, whereas the surrounding stromal tissue or the mammary fat pad is from the mesodermal layer. So they are of different origin. And together, the mammary gland and the stromal tissue make up the breast. Remember, there are two types of mammary epithelial cells. They are myoepithelial cells and luminal epithelial cells, which, if you like, can be further divided into ductal, luminal epithelial cells, and alveoli epithelial cells. So with this in mind, we're going into the early evidence for mammary stem cells. So first, the definition. You have already had 2 lectures of stem cells, and I'm sure you have you have heard about stem cells before. So mammary stem cells are no different from other stem cells that have these 2 essential properties, that self-renewal, and it's able to generate a functional organ. In this case, bear in mind again that stem cells are able to give rise to myoepithelial cells and luminal epithelial cells. So what are the early evidence of mammary stem cells? I'm going to provide 3 lines of evidence from these studies. The first is the generation of mammary glands from a transplantation study. And second is from cell biologist's point of view, is a detection of structurally differentiated cells. And finally, the use of cell surface markers to understand mammary stem cells. The first one, the most important one, is to be able to generate mammary gland from a transplantation study using cleared mammary fat pad transplantation. Just to remind you that in mice, where most of the mammary stem cell studies were carried out, have 10 mammary glands, or rather 10, you can breast or mammary glands, okay? And here is number 4. And this number 4 is of interest for mammary transplantation studies. Okay, with that in mind, what is cleared mammary fat pad? Remember, the breast is made up of the mammary gland and the mammary fat pad, right? So in order to prove that a mammary gland transplanted into the host mammary gland is able to generate a whole mammary gland, then you got to remove all the mammary gland from this host. So people have come up with a clear mammary fat pad technique, which is to clear mammary tissue, mammary gland, from a 3-week-old mice in which the gland has not, the growth of the gland has not reached the landmark lymph node. so that one could make an incision from here before the lymph node and clear the whole mammary gland away. And therefore, this mammary gland fat pad is devoid of any mammary epithelial tissue. So anything transported in would convince you that actually what is transplanted gave rise to the mammary gland. Okay, so this is the early rudimentary structure of the mammary gland. After clearance and following transplantation, you see there is a mammary gland tree there. Okay. Okay, any problem here so far? Yes or no? So you understand what is clear mammary fat pad is where you remove the whole mammary gland from a 3-week-old mice so that this mammary fat pad can be used for transplantation studies to study mammary stem cells. Okay? This just to show you further the growth of mammary gland at different stages. You see this is the landmark lymph node. At every fourth mammary gland, there is this lymph node. And the mammary tip starts here, so it grows that way. This is the very earliest stage. As it grows, it's approaching the lymph node. But at this stage, one could still dissect out the whole mammary gland and to create a cleared mammary fat pad. Where at this point, it's too late because you can't guarantee that you can remove the whole thing. And then when at a later stage in mature adult, the mammary glands occupy the whole mammary fat pad. Okay, so with this technique, people started to demonstrate the generation of mammary glands from a transplanted mammary gland piece. The first report was by this person called Diome, who is the person who established the technique of cleared mammary fat pad. So using this technique, he was able to demonstrate that hyperplastic alveolar nodules transplanted to the cleared mammary fat pad can give rise to mammary tumors. Okay? Why give rise to mammary tumors? Because these hyperplastic alveolar nodules are malignant, and once it's transplanted, then it gives rise to tumors that of certain malignancy. And following that, a number of studies have demonstrated that transplanting a fragment of mammary gland into the clear mammary fat pad can give rise of mammary gland, and you could succeed this by transplanting fragment of mammary gland from mice of virtually any age. And this repopulated mammary gland can be retransplanted many, many times, but of finite passages, presumably because the cells just become senescent like a person. So, but you could do that many, many times. So that is the first demonstration that there are self-renewal mammary stem cells in the mammary gland. But people have always wanted to demonstrate that you could generate a whole mammary gland of this mammary ductal tissue, alveolar tissue, from a single mammary cell. Okay? But it has been really difficult. So until the beginning of this century, all people could do was to using certain technique to label the memory cells, okay, and to show that these labeled memory cells, one labeled memory cells, could give rise to the whole memory glands. So how did they do it? Now, in this study by Colden and Smith, they infected the mice with memory tumor virus. Okay, and then this memory tumor virus will then, at a very young age during nursing, would then be incorporated into the memory gland DNA. Okay, and then they take this memory gland to do transplantation studies. So after they have got the regrowth of the memory gland, they take the regenerated memory gland to study the integration site of the memory tumor virus. So what they found, so the idea is that if this memory gland, the regenerated memory gland, come from one cell, then this tissue, this whole memory gland, should show that there is only one integration site of the virus in its DNA. Okay. How does it work? Do you know Southern blotting analysis? Yes or no? Wake up. Do you know Southern blotting analysis? Yes or no? Okay. So what they do is they so there are some preconditions. So first, they know that the memory gland DNA does not contain sequences that is similar to the memory tumor virus DNA. So if the memory tumor virus DNA gets into memory gland, integrated into a certain site, then you could detect it by Southern blotting analysis. Okay, if you remember how Southern blotting analysis is done, and then you, so they basically design certain restriction digestion, generate different fragment DNA, and then have a probe to detect this band, and they would know whether there is only one band, or two bands, which would result from a DNA that with the virus integrated only at one site, or if they have certain other banding pattern, then they could say this integration is at multiple sites. Okay, so some idea like that. So, but the end story is that they could prove that in a given memory gland regenerated, all the viral integration site is the same. So therefore, they could conclude that most likely this memory gland was derived from one cell that had a single site of the viral integration. Okay, I don't know if you get it, but the idea is you use the virus to mark the DNA, and then you demonstrate that this regenerated memory gland come from just a cell that with one viral integration site. Okay, so that was the earlier demonstration of memory regeneration and memory stem cells. Now, cell biologists usually like to see things through a microscope. So some earlier studies identified memory stem cells through looking at cells under electron microscope. And they proposed that undifferentiated memory cells do not stain well because they contain fewer organelles. So usually the organelles will give you a stronger staining. So these cells are pale cells under light electron microscope. And therefore, these pale cells are likely to be memory stem cells, and they could use this criteria to study, for example, the regulation of memory stem cells under certain hormonal conditions. And they could also demonstrate that these pale cells are depleted in the senescent memory gland. Okay, so this another early evidence of memory stem cells. The third evidence is the identification of classical markers. You know that the progress of hemopoietic stem cells are faster, usually, than the memory stem cells. And then the memory biologists borrowed these markers from the hemopoietic cells, and they started to test these markers in the memory gland. And for a long time, memory biologists believed that memory stem cells have a high SCA1 expression. SCA1 stands for stem cell antigen 1. That is a marker for hemopoietic stem cells. The second one is the presence of site population that efflux Hertz dye. What is site population? This is site population. Have you learned site population? No, okay. So you learned flow cytometry, right? So in flow cytometry, a site population is a population cell that, because of its expression of certain markers, that is away from the main population. So this is the site population. The site population is a population of cells that is away from the main population, shown here. Now, why there is a presence of site population in memory stem cells? They used a DNA binding dye called Hertz dye, I'm sure you heard of. And it's known or it's believed that certain population of the cells express more these drug-resistant gene like ATC transporters, the ATP binding cassette transporters, right? And this protein can expel the drugs, including these foreign DNA dyes. So as a result, so cell population that express high level of APC transporters would have very low level, would not retain the Hertz dye. And therefore, you have a population of cells that contain low levels of Hertz dye, shown here. So this Hertz dye is detected by two different filters. And you see, but nonetheless, it's the lower, very low level of Hertz dye. And that distinguishes this population from the other population. And in the earlier studies, people used to collect these cells and do memory transplantation studies. And they found that this actually gave you more memory regeneration than it would have with the main population, the total population. So people think that memory stem cells also efflux Hertz dye, so it's a site population cells. Lastly, people believed that label retaining memory stem cells is enriched with label retaining cells. I think you have learned this in the last lecture in skin stem cells. So I won't go into detail, but basically label retaining cells are the cells that actually after a thymidine labeling, after DNA is labeled with a tracer, like treated thymidine, this treated thymidine will stay in the DNA of these stem cells for a longer period of time. And the reasons are, some people think that stem cells are relatively quiescent, and therefore the DNA replication is not active, and therefore the label stays. The second reason is the immortal strand theory that says that stem cells will have a symmetrical segregation of the synthesized DNA such that the stem cell will retain the older strand of the DNA during replication, where the daughter cells that is designed to differentiate into other cell types would inherit the newer strand of DNA. So as a result, the older strand DNA obviously have the label, thymidine label, and that way that you could mark the stem cells. Okay, so people believe that memory stem cells also have this label retaining ability. Okay, so pretty much these are the early evidence of memory stem cells. Of course, hundreds of studies have been done. I'm not saying that that's all, but I just give you a flavor of how much people know and what the markers are and how the field has progressed today. So this is pretty much the status until 2006. And then people painted new pictures for memory stem cells. Now, the artists for this painting are a group of people from Australia who published two landmark papers in Nature. One is to demonstrate really that a single cell can generate a whole memory gland. And secondly, they categorized these memory stem cells to say, like, oh, how many memory stem cells in a memory gland, and what are the properties, okay? So I'm going to just show you some of the data, just to give you the real-life situation, what it's like, okay? But before that, you need to know these two terms. The first one is memory repopulation unit, or MRU. You have to remember this not to get lost during the lecture. MRU is memory repopulating unit. It describes cells that can generate memory gland in vivo. So it's usually described as 1 in how many. For example, 1 in 200. It means that in 200 cells, there is 1 memory repopulating unit. Or it could be 1 in 2,000, means in these 2,000 cells, there is 1 memory repopulating unit, or MRU. So obviously, if it's 1 in 200, these cells are more enriched with memory stem cells than in 1 in 2,000, right? So you have to understand this concept. So this is MRU. And the second concept is memory colony forming unit. Memory colony forming cells. So this term is used to describe memory progenitor cells that can form colonies in low-density adherence culture. So basically, MCFC describe cells that can form colonies or mammospheres in a cell culture condition. Okay, got it? MRU is more important because I'm going to talk about it right now. Okay, so as I said, that this study, the first study, demonstrated that they can generate whole memory gland with just single cell. At that time, it was really, very, very exciting for the field. And what they did was to sort the cells by flow cytometry and then enrich the cells with certain markers. Okay, so first, they use certain markers like 45, 119, and 31. You don't need to memorize it, by the way. So these markers identify, for example, 45 and 119 identify hemopoietic cells, blood cells, Right, 31 identify endothelial cells, the blood vessel cells. So they first sorted the cells and removed all these cells. And these cells, after remove these cells, the cells are called Lin negative. Where is Lin negative? Okay, just remember, I can't remember where the word is. Oh, here, okay? And then they got this population. After that, they tested two markers. These are CD24, that is a cell surface marker. Obviously, you can label this. And CD29, which is integrin beta 1. And so I won't go into detail unless we want me to explain. I'll explain to you later. Basically, this is the population that labels high level of CD24 and CD4 positive, CD29 high. So because the whole population is CD29 positive. But at this line above, and they define this as CD29 high. Okay? And these dotted lines are just isotope control. And then after sorting, basically they collected this population that is CD24 positive and CD29 high. And they transplanted these cells into mammary gland. And they got MRU, memory repopulating unit, of 1 in 4,900. So that is not very impressive. No. I was wrong. So the Lin negative population, that is the one that removed all the hemopoietic cells, the MRU is 1 in 4,900. Okay? But the MRU in Lin negative and C29 high, C24 positive was 1 in 64. So, which means this CD29 high, CD24 positive are enriched with MRU and hence memory stem cells. And later on, they do dilution. So such that per injection, there is only one CD24 high, positive and CD29 high memory cells. And they want to see whether they could generate mammary gland with just one single cell. And they succeeded. So these are the mammary glands whole mount images. Show, and these glands are labeled with LAG-Z, okay? And they show that at different stages of memory development, that these glands were generated from injection of a single memory cell. And then they further label this at showed image of higher magnification, that essentially to tell you that this single cell could give rise to both the luminal epithelial cells, shown here, and myoepithelial cells, shown in here. Okay, so this one cell can give rise to lineages, luminal epithelial cells, and myoepithelial cells. Their success rate is 8 out of 68 injections of single cell that gave rise to mammary gland. Okay. So they claimed that the first time they were able to generate the whole mammary gland with a single cell. So that was remarkable. But then in science, you got to take consideration what is known in the past. You can't just say, okay, that's it. But are your studies consistent in agreement with others? If not, why? If it is, why? Okay? So in this case, They have identified new markers with CD29 and CD24. But these were never regarded as the bona fide stem cell markers. So they got to go back in history to check the other markers that people actually believed in, right? So they try to see whether this stem cell population were enriched for long-term label retaining cells. Okay, so they injected BRDU, which is analog of thymidine, and then they could detect it weeks later. Okay, so when they first injected, and they could see the BRDU positive cells in both CD29 low and CD29 high. Remind you that CD29 high are supposed to be the stem cell population, right? So there were no difference at the beginning, at the time of injection, which is okay, control. And then after 7 weeks, they showed that CD29 high cells have much more BRDU returned cells than the CD29 low cells, which is not the stem cell population. So it's good. It's in agreement with the general concept that memory stem cells is enriched with label retaining cells. Okay. Then there are 2 others I told you about. One is the SCAR1. The second is the site population. Remember? Okay, so basically what they show that SCAR1 staining shown in here, so this 63% population is SCAR1 high population, does not contain this marker that CD29 high and CD24 positive. So meaning that now the marker 29 and 24 positive cells do not have high SCAR1. So therefore, SCAR1 may not be the right memory stem cell marker. And this is just to mark that the SCAR1 positive high cells. Okay? And then next is the site population. So they wanted to see whether CD29 high, CD24 positive cells contain more site population, because this is supposed to be the cells that express high levels of APC transporters and therefore were able to expel the label, the Hershey dye, and therefore you will see more site population at this corner, right? The truth is no. So in this population cells, there's only 0.1% site population, whereas in lean negative population, there is 1.2%, that is 10 times more. So this population is not enriched with site population. Okay, and then they further verify that this site population is the Hershey-Dye effluxing population. So they used verapamil drug that is a APC transporter inhibitor. So once they use this drug, they could remove this population, proving that this population is indeed the site population that can efflux Hershey's dye. Okay, so far? Yeah. So these are the studies how they try to reconcile with the known data. And so how do they explain that why the data agree with previous one or not agree with previous one? They think that SCAR1, why their population, the stem cell enriched population, do not express high levels of SCAR1 because they used freshly isolated memory cells for injection, whereas the earlier studies cultured these cells for several passages. And they demonstrated if they culture these cells for several passages, then they could increase SCAR1 population dramatically, and therefore they believe that while the earlier studies claimed that stem cells enrich with SCAR1 because they have passaged their cells in the culture before they inject into the memory fat pad. What about the E-flux Hutchins dye? They say that in the early studies, they use a very high population of SP1 cells. And therefore, that when you use a very high population, then there is high chance that one could generate memory glands. And also, they found that SP cells is enriched in CD24 positive, CD29 low, and CD49 low cells. So these are progenitor cells. So, so basically, they say why their studies their stem cells don't have the site population because they do not enrich for this population. But other studies enrich this population. Remember I told you earlier that they collect this site population through injection. In order to have enough cells, they actually collect a lot of SP cells. And they say that is the reason why they could demonstrate memory regeneration with SP population, because they use a lot of cells, and these cells actually is sort of contaminated with some real memory stem cells. Okay. So this is the first study. Basically, what they did is A, identify markers. B, they demonstrated they could generate memory gland with a single cell, and they also discussed their data with known data and whether the existing markers still stand or not. Okay, and then so the second paper I'll go through briefly. They just simply characterize like how many MRU, how many stem cells in memory gland. They say that using certain conditions, they can demonstrate that it's 1 MRU per 1,400 cells. That is the total memory gland population, not enriched for any markers. Okay, and so in that case, that would be 1,400 stem cells MRU per gland. So meaning there are 14 memory stem cells per memory gland. And they also demonstrated that another marker, CD49F, which is integrin alpha 6, high cells contain high MRU. So in addition to 24, 29, and there's a 49F. So you got to remember. And if further 49F high CD24 positive can even increase the MRU further. And what about the mammosphere formation in a cell culture condition? So there is 1 MACFC per 63 cells, which is much higher. Because remember earlier it's 1 every 1,400, or 30,000 MACFC in the whole gland. And they also showed that there are two populations that can form different types of mammospheres. Shown in here. If it's CD24 positive, CD49F low, the cells form a hollow mammosphere. And then if all this high, then the cell will form a solid mammosphere. Hollow means you see if you make a section inside that it is hollow. But this one, if you make a section, then inside there are a lot of cells. So this is the population that is enriched with memory stem cells. So they form different mammosphere. And why they want to do that? Because then if you have these characteristics, you could study memory stem cells under in vitro condition for various applications, because it's not always easy to study in animals. Okay? So which is to say that this solid mammosphere is enriched with memory stem cells. And then you could study this solid mammosphere. And they also suggested that this hollow mammosphere are formed by memory progenitor cells instead of the memory stem cells. Okay, we have a break here. And come back in 10 minutes at 9:30. Okay, 9:30, let's start again. Okay, so in the second part of the lecture, I'm going to just to continue on the new definition of memory stem cells and differentiation hierarchies. And after that, I'm going to talk about memory stem cell niche and breast cancer stem cells. So you need to know two things to understand the later part of this lecture. The first is that I repeat, memory gland contains two lineages. These are the luminal cell lineage and myoepithelial cell lineage that give rise to myoepithelial cells and luminal cells. Okay? So following the 2006 studies, a number of studies were carried out to characterize the memory stem cells and their differentiation hierarchy. And then they come up with this model in 2009. I put it as a classic model of differentiation hierarchy within the memory gland. So you see a number of markers. Be assured that you don't need to memorize all these markers, and you don't need to memorize especially the human stem cell markers. Okay? So what is the model for memory gland differentiation hierarchy? So what is this? If you look at the blue, which represents the mice memory stem cell markers, is that the stem cells express these markers, and it gives rise to a common progenitor cells, which then is responsible for the generation of luminal progenitor, alveoli progenitor, which are both luminal cells, and myoepithelial progenitor. Okay? And then the luminal progenitor will give rise to both ductal cells and luminal cells. But this alveoli progenitor that comes from the common progenitor also give rise to alveoli cells. Okay? And it's said that alveoli progenitor can also generate myoepithelial cell progenitor, but it's a dotted line and therefore is questionable. So the question is, do memory epithelial lineages come from a single progenitor or not? Now this question was answered in an elegant study published at the end of 2011. You can look up this paper if you're interested. Now this person, I put his picture there because he was named as Nature's magazine, Nature's Top 10, who made a difference in science in 2012 for his group study on genetic lineage tracing studies of the memory stem cells. And so he was named as a cell tracker, because he used this study and he used this technique and did some elegant studies on lineage tracing. Have you heard genetic lineage tracing before? Yes or no? What is genetic lineage tracing? Yes or no? You have? Okay, excellent. So I don't need to explain. So his group wanted to know exactly under in vivo condition without transplantation whether the memory stem cells come from one progenitor cell or different progenitor cells. So their studies were based on the understanding that basal cells, so you have epithelial cells. Epithelial cells consist basal cells and luminal cells. Basal cells in mammary gland also called myepithelial cells, right? So the basal cells or myepithelial cells express CD14 and CD5. These K14 or K5, they are cytokeratin markers, which are the intermediate filaments in the cells. And the basal cells and luminal cells express different markers. But they are not cell surface markers, by the way. Okay, they are cellular proteins. So K14 and K5 are expressed in basal cells, whereas K8 and K19 are expressed in luminal cells. So then they generated mice model where they can genetically label these lineages and then follow their progenies. I'm not going into the details of this genetic manipulation. So basically, they can do the conditional on-off a gene. In this case, is the yellow GFP protein, YFP, so that they follow, once the cell is labeled or switch on YFP gene expression, then they trace these YFP cells after a certain period of time to see whether the YFP cells that goes to luminal lineage or myoepithelial cell lineage. Okay? So this is basically a summary of the existing knowledge that in the earlier mammary gland, the rudimentary mammary gland in embryo, all the cells, all the memory cells express K14. But the center part of the cells, some of the center part of the cells also express K8. Okay? So needless to say, the mammary gland come from this class of cells because these are the progenitor cells during embryonic development. So all the mammary gland must come of these cells. But where are these markers go? So they could label early stage and see where they go. And they find that K14 cells are present only in the myepithelial cells or the basal cells, whereas K18 mostly or almost entirely in the luminal cell. And then they also mark K5 here and K19, but some luminal cells also express both K8 and K14. Okay? This is at birth and is practically similar except for the fact that basal cells express exclusively K14 and K5, whereas K8 and K19 only express in the luminal cells. And there is no hybrids, like both K14 and K8, there is no such a thing in adult virgin mammary gland. So this is the basic knowledge and summary of the findings. So the real data is here. So first they used immunostaining to see the staining of K14 and K8, and practically they demonstrated this. Okay, that during in the rudimentary mammary gland, all the cells express K14, but some cells in the center of the mammary plate express K8. Then at this stage, they switched on YFP expression by using doxycycline, so that this YFP is under the control of K14 promoter. And then they see whether this YFP cells is present in the luminal lineage or myoepithelial lineage. After, when the mice were 5 weeks old, they did the treatment at E17, okay, and tested the marker expression at 5 weeks old. What do you see? You see that all the cells express YFP. And the K5 with basal cell marker is in the basal site, okay? But if you co-stain the K8 with YFP, you also find that they overlap, which means that these cells, K14 cells, gave rise to both the myoepithelial lineage and luminal lineage, because all the cells express YFP. And then they also tested if these YFP cells are particularly enriched with the known stem cell marker, that's CD24 and CD29. And they found that not really. So YFP cells are CD29 positive or CD29 high. And in these two populations, so the one in light blue are supposed to be the cells that enriched with stem cell population. They are yellow, okay, express YFP. So are those cells that are not supposed to be stem cells. They are also yellow, just to show that the yellow cell give rise to all the cells, okay? So the conclusion is that all memory epithelial lineages are derived from embryonic K14 expressing progenitors at this stage, if you mark the YFP cells at embryonic stage, which makes sense because after all the cells express YFP, and the mammary gland must come from here, so they have to be all yellow, express YFP. Then what about during memory expansion and maintenance in adult? Memory expansion and maintenance occurs, as I said, that during estrous cycle, when the mammary glands undergo growth, apoptosis, and also during pregnancy, which is more obvious, okay? So they then switched on YFP in mice during estrus after it's sexually matured. Then this is about 4 weeks old, and then they feed the mice with tamoxifen or doxycycline to switch on the YFP. First is the K14. Okay, you see at 5, I think it's 5 days, they put it 1 week. So let's say 1 week after the treatment, switching on the YFP, these cells are in the basal layer. K14 positive and YFP positive cells are in the myoepithelial cell population. Okay? When co-stain K8 with YFP, you see there is no YFP and therefore no K14 cell-derived cells in the luminal population, because the YFP is present only at the basal population. And so that is okay, that is after 1 week. 10 weeks later, when the mammary gland have undergone, cyclic changes, maintenance, regrowth, and it's still the same, that K14 is present exclusively in the myoepithelial cell layer, and it does not co-localize with K8 with the luminal cell. In other words, K14 cells or YP cells does not give rise to luminal population. Okay? So K14 stem cells give rise only to myoepithelial cells. And the same thing applies to K8. K8 positive stem cells, when they did the genetic tracing, gave rise only to luminal lineage. And this just repeats what they said, except that they label the YFP goes with the K8 positive cells. And after 10 weeks, the luminal cells express K8. And luminal cell, these K8 positive cells are not present in the myoepithelial cell population. And with that, they proposed a revised model for mammary gland differentiation hierarchy. So they suggest that at the embryonic stage, the stem cells, the mammary stem cells, are bipotent that can give rise to both luminal and myoepithelial lineage. But after sexual maturation, during mammary gland expansion and maintenance, CK8 positive luminal progenitor cells is responsible for the luminal cell lineage, both ductal and alveoli luminal cells, whereas the myoepithelial cell progenitor is responsible for the myoepithelial cell lineage. But this is, again, in disagreement with the earlier model. And the author suggested that why this is different, because their study was in vivo situation where they did not really take out the mammary cells and then transplant into the mammary fat pad. So that forced growth actually gave you an artifact that if you inject these basal progenitor cells into the mammary gland, somehow the condition can force it to become bipotent. Therefore, it can generate both the luminal and myoepithelial lineage. And I've summarized here to highlight the difference in the classical model and the revised model. I shall not go over here. Okay, so last two sections. The next is mammary stem cell niche. Now, I think you have learned about this in your skin stem cell, am I right? So there is a microenvironment that supports the maintenance and expansion of any stem cell, including the mammary stem cells. And for mammary gland, the mammary stem cell niche is in the mammary fat pad. It's hardly surprising that mammary fat pad is essential for mammary gland development, because if you inject mammary stem cell into any part of the skin, they're not going to form mammary glands, right? Despite of the fact that the seeming content of the mammary fat pad is no different from other connective tissue or fat pad, because the mammary fat pad is, modified connective tissue, which means it's connective tissue, but it has a lot of fat cells in there. And at this point, we don't know why it's special, but you need this mammary fat pad, this particular microenvironment, to, maintain the mammary stem cells and to support the growth of mammary gland. So it must be very important, very special, that we get to understand what is the special factors there that is needed for mammary gland development. So the mammary fat pad, together with mammary epithelial cells, actually can redirect stem cell feed. For example, it can force other stem cell types to become mammary gland. The first one is from the study of spermatogenic cells. So these people are crazy. They wanted to know whether the mammary fat pad is so special and together with mammary epithelial cells that they can convert spermatogenic cells into mammary cells. And they have succeeded. Show you in here. So what they did is to, of course, these cells are labeled with LAC-Z. They mixed spermatogenic cells with mammary cells, properly enriched with the markers, mammary stem cell markers, transplanted into the mammary gland, and after one round of pregnancy, they found that male cells are present in the mammary alveoli, shown you here. You see there are XY indicating the X and Y chromosome, X in red and Y in yellow. Okay, so you see in this acini, there are cells made up of XY chromosome, there are cells made up of XX only. So what is the study trying to convince people is that the mammary fat pad is this mammary stem cell niche, that it can force the other stem cell to change its fate, okay? And these cells adopt mammary epithelial progenitor properties. The second demonstration that the mammary microenvironment or mammary fat pad is so important is that they were able to force the neural stem cells to become mammary stem cells. Simply put, that they labeled, again, the neural stem cells with LAC-Z and transplanted together with mammary stem cells. And you see these neural stem cells, they are blue, become generated mammary gland, shown in here. Again, this photo is to show that the neural stem cells actually can contribute to both the myoepithelial lineage, they stain this a little bit brown, this is the myoepithelial cells, and of course the luminal epithelial cells. Okay, the last section is on breast cancer stem cells. What are breast cancer stem cell hypotheses? It is that breast stem cells and progenitor cells are the initial targets for malignant transformation. In the tumor, there is a population of cells that retains key stem cell properties. That is cell renewal and give rise to a new population, tumor generating and self-renewal. So how did it first come about? The first report or suggestion of cancer stem cells come from the studies by John Dick and colleagues. And they described a reproducible way of enriching cells with tumor-initiating activity, and therefore ruled out the stochastic model that is not random mutation that generates the tumor. And the acute myeloid leukemia initiating cells not only were able to differentiate and proliferate, but also had the capacity for self-renewal in vivo. So this is the key attribute of stem cells, and that obviously has a lot of attractiveness if tumor are indeed from a small stem cell population that one could target it, right? So the real breast cancer stem cell demonstration come from the study in 2002. That's about 10 years ago, in which they could demonstrate that certain markers identify breast cancer stem cells, and these cells can have the property of self-renewal, and they give rise to a population of tumor cells. So what I want you to get from this table is a bit complicated, is that when they get the human tumor mass, they were able to generate tumor in immune-deficient mice, shown in here. If they enrich, here is CD44 positive and CD24 positive, there were very little tumor formation, okay, at the bottom. But if they enrich the cells with CD44 positive and CD24 negative, then they could increase the tumor formation significantly, even at lower cell number. And by adding on one more marker, that is the epithelial cell antigen, then they could generate even more, generate tumor with even less cells. So 1,000 cells could give rise to a tumor. And so once these are passaged, so you get the tumor, grow in mice, and take from the mice, and then disperse it and count it and re-inoculate into the mice. So then your passage is 1. So mouse passage 1, meaning that is after the first tumor formation that re-inject into mice. And then they studied whether the marker CD44 positive, CD29 negative, ESA positive have a greater capacity to generate tumor. And the answer is yes. So I shall just omit this and just to show you that with as little as 100 tumor cells, it can give rise to tumor. With 200, the success rate is 100%. So these markers are cancer stem cell markers. And the same thing can be demonstrated with further passages that I'm not going into. But furthermore, what they could demonstrate is this cells that are expressing the CD24 negative and ESA positive cells that can give rise to other population of cells. So you see, they mark these cells, CD24 negative, shown in here, inject into mice, and then they could generate a mixed population, yet still retain some cells in this population. The same is true with CD44 positive and CD24 negative. So they marked CD44 positive here, but CD24 negative here, right? And give this population, and then it generates a mixed population, yet still retain certain population expressing CD24 but not CD24. So these are the demonstrations of memory cancer stem cells. So what is the point? The points are that whereas the majority of the cells in the cancer mass do not initiate tumor, because you need so many more cells to generate tumor if you don't enrich the cells with these markers. Minority of CD44 positive, CD24 low, ESA positive population initiated tumor formation. And these cancer-initiating cells were also present in serially passaged tumors, like I showed you. And together with phenotypically diverse population of non-tumorigenic cells, and therefore these markers are candidate markers for breast cancer stem cells. Okay. Now, ever since then, people went crazy about finding breast cancer stem cells because it's very useful if you find them. Apart from this, we want to find better ones. Then that can be a drug target, right? So a lot of studies have gone into looking for more breast cancer stem cell markers. And one of them stood out. That is the aldehyde dehydrogenase. That is not a cell surface marker, but one could, have a reporter gene to detect their presence in the cancer cell population. So what this study showed that in 23 out of the 33 breast cancer cell lines, contain the population that express this aldehyde dehydrogenase and tumor-initiating cells. And these cells, actually, the ones that express high level of ALDH1 are those that are more aggressive and more metastatic, as is shown in here. So they tested 3 cell lines that either express the ALDH1 alone or together with IO8, and they found that the ones that do not express ALDH1 have lower invasiveness in vivo metastatic model, whereas those expressed have significantly high invasion rate, and together with IO8, these cells are more malignant, and therefore they suggested that these aldehyde dehydrogenase marks invasive tumor-initiating cells. Okay. So as I said, that cancer or breast cancer stem cell theory is very attractive because people, obviously, if that is true, you identify the marker, you target this marker so that you can prevent these cancer stem cells to progress to generate tumor. So that is one point of therapeutic targeting. Even after the tumor formation, if you could target this tumor, then because these tumors are more aggressive, tumor initiating, and then you could prevent tumor growth and tumor metastasis. Whereas if you just left with these non-stem cell tumor cells, they are more easier to deal with because they are less malignant and they can be more easily eradicated with the conventional chemotherapy that just kill the proliferating cells. Okay. The point is life so simple? Obviously not. And cancer stem cell, of course, is like many other fields, is a controversial field. And there are believers, non-believers. I think one of the studies provided evidence that actually the so-called breast cancer stem cells or cancer stem cells may be just the aggressive tumor cells. They demonstrated that actually the tumor formation potential is a function of the tumor microenvironment. It depends on whether the host is a permissive host to allow the tumor to generate. So what they did is to compare the tumor formation potential of these melanoma cells in 2 different hosts. One is the usual SCID mice, with NOD stand for obese diabetic mice, because these mice will develop diabetes at an early age. So the usual one, and the one that also is interleukin-2 receptor gamma now. This made the mice more immunodeficient, and therefore has more impaired immunity. Okay, and they found that the same tumor cells can form tumor hundreds, thousands of times more than the counterparts, the NOD skip mice. Actually, no, it's like 20,000 times more. So which means it's the same cell, and just because of different microenvironment, and then the tumor initiating potentials are totally different. And therefore, so-called cancer stem cells are not valid. If they are cancer stem cells, then they should have the same tumor generating potential in all the whole cells. Okay, so that is the idea that the tumor-regenerating potential actually depends on the microenvironment. And also, they couldn't find any markers that can distinguish between the tumorigenic and the non-tumorigenic melanoma cells in these two different hosts. Okay, it's not that the IL-2 gamma now host actually cultivated a certain cancer stem cell phenotype. No, there wasn't. Of course, it's a question whether the finding is extendable to breast cancer stem cells. So here comes the model, which support the idea that there may be not such a thing called cancer stem cell. Maybe there are tumor-initiating cells, but these are just aggressive malignant cells that are selected during tumor progression. So this paper argued the case with the development of marker, the hallmarks of cancer development. So their theory is that these normal cells, or tumor-initiating cells, can become malignant upon these initial genomic events that is listed here. Once it acquired this capability, then it gave rise to stem cells. And these stem cells, then under certain selection pressure, if they have developed these hallmarks of cancer with all this angiogenesis, invasion of EMT, and with the expression of these genes, genomic instability, pro-inflammation, and there is another thing called deregulating cellular energetics. And this selection pressure force the tumor cells to become more malignant and invasive, and they are able to generate a larger population of cancer cells because their ability to deliver the proliferation potential, and the cells were able to have acquired ability to metastasize to the other sites. So this is the status in the cancer stem cell field. To summarize, you're starting to pack up. Okay, I leave you to read this summary then. Okay? It's okay? Okay, I go through this. So basically, what I told you, that there has been a paradigm shift in the understanding of memory stem cells in the past decade, since 2006. And first, people identified the markers. They are still valid today. And then they figured out the differentiation hierarchy with some revision. And the traditional markers, such as SCAR1 and Psi population, no longer valid. And these are the new markers that I hope you can remember. the CD29, 49, 24. These are the mice stem cell markers, remember? And whereas in human, it's another set, CD49 epicam and CD24 negative. Okay? So I want you to at least remember these markers. And recent genetic tracing studies revealed that the myepithelial and luminal lineages in adult mice are maintained by unipotent memory stem cells. This is important because in the past, people always believed that if cancer comes from the stem cells come from the basal lineage, and therefore one should target the basal lineage cell only. Whereas now we know that both luminal and myepithelial lineage progenitor cells can give rise and generate mammary gland. And therefore, that just opened a door for further study into these two populations of cells for their tumor initiation or generating potential. And memory microenvironment can redirect the cell fate of other stem cells. And breast cancer stem cell theory is being redefined. We're still waiting to see what is to come up in the next 10 years or so, which I believe you guys will play a very important role in the cancer stem cell research. Okay, that's all for today. And remember to send me questions for the tutorial.